This final screencast on muscle physiology is going to focus on this contractile cell of the human body. Hopefully you can all identify it. You can just pause slightly if you want. Basically, this is smooth muscle. Hopefully nice and characteristic, no stripes, therefore have the smooth appearance. What we'll do in this final screencast is talk about the properties of smooth muscle compared to cardiac and skeletal muscle, and compare and contrast some of the proteins that are involved in excitation, contraction, coupling. What will probably a take-home message from this is going to be probably that we'll see the proteins that are involved in EC coupling in smooth muscle are somewhat different to those seen in cardiac and skeletal muscle. So just some properties. Smooth muscle is the muscle's internal organs, particularly probably the gastrointestinal tract, the renal system, or the urinary system, but also, of course, for blood flow, smooth muscle surrounds our blood vessels, and we can either get vasoconstriction or vasodilation to control the flow of blood via the control of smooth muscle contraction. Smooth muscle is so named because it has no visible stripes or striations. And again, hopefully you realize now its contraction is involuntary. It's not neurogenic, it's myogenic. Smooth muscle fibers are spindle shaped and they're joined by specialized junctions. Again, we'll probably see this over the next couple of slides. Adapted for different functions. So maybe a little bit like we were talking about the motor unit in skeletal muscle. The motor unit could be small for fine control of a small organ such as the eye, or it can be very large when we're looking at the contraction of large muscles, and I think the example used was the quadriceps in the leg. Similarly, smooth muscles can be single unit, again coupled by gap junctions, things like gastrointestinal smooth muscle, or multi-unit, primarily under neural control, but essentially you don't need a neural input in order to activate the system. Vascular smooth muscle and ciliary muscle and the iris of the eye are examples of multi-unit control, innervated by the autonomic nervous system, but essentially myogenic in nature. So these are the characteristics of the smooth muscle, and again, maybe we'll just focus in on some similarities and maybe some differences when we come to them. So in terms of sliding filament and actin and myosin, essentially similar in that actin and myosin are going to slide against each other in order to let muscles contract. The thing that is different is probably the ways in which we control, or which calcium controls the system. Again, we'll see this as we go through the next few slides. Smooth muscle, as we said, also uses actin and myosin for contraction, but these are not arranged as regular sarcomeres, I should say. Regular sarcomeres are a feature of certainly skeletal muscle, as we've seen before. So this is maybe the main difference here. Contraction differs from skeletal muscle and cardiac muscle, in fact, in that smooth muscle uses trypomyosin but lacks troponin C, and instead it uses another calcium binding protein called calmodulin. So calmodulin activates what we call myosin light chain kinase, and this phosphorylates the myosin heads, again to act via the sliding filament system. There are no T tubules as we see in skeletal muscle and cardiac muscle. Contraction develops more slowly but tends to last longer than skeletal muscle. ATP usage is much less, and slower than we see in skeletal muscle. Again, hopefully this becomes clear over the next few slides. Excitation in terms of the neural input is more complex and diverse. What we'll see is that, well, there are a number of different types of action potentials that are used. They tend to be much less well defined than the very clear different phases that we showed you for the action potential in the cardiac system, for example. Action potentials can be longer. You see they can be, again, longer but varied between 10 and 50 milliseconds. And it involves the influx of both sodium and calcium. And again, this can differ between locations. Also driven by calcium second messengers, so some neurotransmitters that release calcium from the sarcoplasmic reticulum also allow us to control smooth muscle excitation to a much greater extent than is seen in skeletal muscle and cardiac muscle. Anything that can increase calcium, for example, hormones, and also things like gasotransmitters, nitric oxide, all implicated in the control, the fine-tuning of smooth muscle excitation in the gut and the other organs, much more than we see with skeletal muscle and cardiac muscle. So again, maybe the idea about smooth muscle excitation is that it's less regularly controlled than skeletal muscle and cardiac muscle in particular, which needs to be synchronous. We can use, we can digest our food at relative leisure by exciting smooth muscle. It's not as critical to life to do that as to control skeletal muscle in a well-defined synchronous way. So yeah, again, this is another schematic of how excitation, contraction coupling works in smooth muscle. Again, we're going to see that there are a number of different proteins in smooth muscle contraction. So essentially, again, we have probably an action potential coming in and calcium coming down its concentration gradient. We still get this calcium-dependent calcium release from the sarcoplasmic reticulum, but this is where things start to change. So in smooth muscle, there are two different proteins that are involved, as we said on one of the previous slides, calmodulin and myosin light chain kinase. So the idea is here that calcium binds to calmodulin, probably as its name suggests, it's a calcium binding protein, it's modulated by calcium. Calcium calmodulin in a complex activates myosin light chain kinase, and again, probably as the name suggests, myosin light chain kinase is responsible for phosphorylating the light chains present in the myosin heads, and this is the thing that increases myosin ATPase activity in smooth muscle. So you can see, although we've still got activation of myosin heads, we've still got this energy dependent process. The proteins that are involved in smooth muscle contraction are different to the ones involved in skeletal muscle and cardiac muscle contraction. Active myosin cross bridges are released so that we can slide actin and myosin across each other. So again, it works by the sliding filament module. It's just that the calcium regulation is slightly different. Again, when we want to cease smooth muscle contraction, we need to get rid of the calcium. So free calcium is decreased, again, when we pump calcium out of the internal environment back into the stores. And again, maybe the proteins that are involved in here are similar to SERCA, they're ATP-driven pumps. We don't actually know the exact molecular composition of these pumps, but they're similar in nature to the SERCA pump. Myosin phosphatase, an enzyme, removes phosphate from myosin, which decreases myosin ATPase activity. Less myosin ATPase results in decreased muscle tension. So similar, but slightly different to the excitation contraction coupling that we've seen in skeletal and cardiac muscle, which is much, let's say, more well defined and well controlled. So probably again, we started off with a big slide at the beginning which showed you the difference between the three muscle types, skeletal, cardiac and smooth, and we're almost coming back now to a slightly smaller version of that type of table. as we said, we don't expect you to know this in a great deal of detail, but by now, listening to the 5 podcasts, you should appreciate that there are similarities between skeletal, cardiac and smooth muscles, but some differences as well. The way that excitation is coupled to contraction is slightly different. Obviously, in the skeletal muscle, we've got our neuromuscular junction, we've got release of acetylcholine. Cardiac, we have more, it's the one system where we have extensive use of gap junctions. Again, let's look back at that 4th screencast if you want to see how the gap junctions work. The action potentials come in spikes. Here they're more wider, they've got a plateau because of the calcium, L-type calcium channels. And actually, if we showed you the action potentials for smooth muscle, they're actually very varied. They're not this classical shape of the cardiac action potential, and indeed the nerve action potential that you see at the skeletal muscle. The calcium sensors are similar between, or the same between skeletal muscle and cardiac, but remember there are certain isoforms of troponin, C, I and T in the cardiac muscle. Here we use calmodulin. This is probably, again, similarities in the way that excitation is coupled to contraction. The termination is different. So acetylcholine is broken down by acetylcholine esterase. Here we tend to use the cardiac action potential a little bit more to control contraction. And here we use myosin light chain myosin light chain phosphatase to inhibit the sliding filament in the myosin interactions with actin. The duration of twitch, the movement of the muscle, is different, if you like. Remember, we've got red and white fibers here which control fast and slow twitch. Cardiac action potential is slightly wider. Smooth muscle, again, is quite different. The reason that we don't show you a typical action potential in smooth muscle is because there's probably no such thing as a typical one. They're very varied. And again, maybe just to finish off metabolism, we've shown you that red and white fibers use different forms of oxidative phosphorylation and glycolysis. Here, for cardiac and smooth muscle, they're both oxidative processes. And again, this is really just to go back to that table. As I said, we've picked out some important parts here. This was a table that I showed you at the beginning. So hopefully now, when I pointed to things like troponin and calmodulin, and hopefully back at the beginning, maybe you didn't know what myosin light chain kinase was. Now you know what it is. So there are similarities, but clear differences between the ways, the proteins that are involved. But essentially, we have calcium-dependent processes that lead to the coupling of excitation to contraction in all systems. So let's summarize here. In the heart, this is obviously summarizing the cardiac and smooth muscle podcasts, screencasts, Screencast 4 and Screencast 5. In the heart, pacemaker cells in the sinoatrial node set the rate of cardiac muscle contraction, the pacemaker. In smooth muscle, contraction is initiated by events that allow influx of calcium. Again, as we see, calcium influx is very important for all muscle contraction. Skeletal, cardiac and smooth muscle all use myosin. They all use actin, they all use ATP, and they all use calcium. Muscle types differ in the source of calcium, the arrangement of actin and myosin, excitation, contraction coupling, and the regulation of contraction. In particular, remember, smooth muscle is the one that uses calmodulin and myosin light chain kinase, whereas skeletal and cardiac muscle use troponin. Okay, 15 minutes, that's a good place to finish here. The activities that are associated with these screencasts are embedded into the screencast, so there's no separate activities for these. But again, have a look back, and be able to make sure that you can compare, contrast skeletal, cardiac, and smooth muscle contraction.